Today I'm going to talk about BD solution for cervical cancer screening using both BD SurePath liquid-based cytology and BD Onclarity HPV assay. So just wanted to start by saying that BD offers a continual partnership with the customers in India in their cervical cancer screening program. So we offer a complete solution for the cervical cancer screening for both LBC as well as HPV. Our BD Onclarity HPV assay is FDA approved for extended genotyping. We do have a number of international speakers for cervical cancer screening updates and best practice sharing. As some of you are aware and may have attended the We Hope seminars and webinars that happens every year, we are heavily involved in continuous CME training and education program for the laboratories, as well as for the gynecologists and pathologists. And that we offer an excellent cytology and HPV DNA testing solution for cervical cancer screening. So let's start with sample collection methods. So we have a BD SurePath collection vial that contains 10 mils of 24% ethanol-based preservative fluid. When the sample is taken, the sampling device or the brush head is broken and left into the vial, and the whole vial, which includes the sampling device or the brush head, is sent to the laboratory. The reason for sending the whole brush head is to make sure that 100% of the sample that is collected is sent to the laboratory for processing. And once the LBC slide is made and processed, there is enough sample left inside the vial to do ancillary test, for example, HPV test. We have three types of sampling devices. We have Rover's cervix brush, which is most commonly used by the gynecologist. We have the combi brush, where the middle part is extended, and it is most commonly used for women who have dry cervix or women who have had previous endocervical abnormality, and the gynecologist or the pathologist wants to focus mostly on endocervical cells. And the third option is the Rover's endocervical brush and spatula combination, and it's used together and sent in one SurePath vial to the laboratory. So our collection method is standardized, and it is 3 simple steps. Collect. So we collect, the sample is collected from the woman. Once the sample is collected, it is dropped into the SurePath vial. So the sample head is dropped into the vial. The vial is then secured, and then it is sent to the laboratory with the sampling device inside the vial. And this ensures that whatever is collected from the woman, 100% of the sample collection is sent to the laboratory. So BD also has 2 different methods, semi-automated as well as automated solutions, depending on the volume of samples you're receiving in your laboratory. The first one is BD Direct to Slide or DTS method, where the slide is secured in between a holder, which is a reusable holder, so you can wash it and use it in your laboratory, and settling chamber. And once it is secured, the technician will put in the sample and process the slide. And I'll go over it with you how the processing is done in my next slide. Then we have the automated method, which uses the BD Totalis SlidePrep. And this processes as well as stain the slides. So the slides are made here and stained here. And this is where we keep all the reagent and the wastes come off. And again, I will go over it in detail in my later slides. So first, what is BD SurePath Direct to Slide? It's a simplified innovative LBC method, which is designed for laboratories with low to mid-volume workload for cervical cancer screening. And all you need for this is the sampling device and the SurePath vial to put the sample in. And in the laboratory, the holder, the slide, settling chamber, and some reagents. So the workflow for this semi-automated method, DTS or direct to slide, is once you have received the sample in your laboratory, you vortex the sample to homogenize it. And then the technician will make the holder, prepare the holder for to put in the sample. So this is made by securing the slide in between the holder and the settling chamber. And when this is done, you put in 1 ml of density reagent and you layer it with 2 ml of sample on top of it. The entire thing is then put into the centrifuge and centrifuge for 2 minutes at 200 G. And then after 2 minutes, it is inverted and left aside for 1 minute at a slant position. And after 1 minute, your slide is prepared. You don't have to do anything except to stain it. So you can either do manual staining or put it in a staining machine to stain it and cover slip it. And then your slide is ready to be screened under the microscope. So for the automated method, we have BD Totalis SlidePrep. And the BD Totalis SlidePrep is an automated instrument that processes as well as stain the slide. So you don't need another separate stainer to stain the slide. It is ideal for laboratories who have constrained with space. And once they have purchased this BD Totalis SlidePrep, there's no need for a different stainer at all. They can stain the slides in here. It also has a touch screen attached to it, so it's quite easy to use because the touch screen guides you all the way during processing, staining, maintenance, changing of the reagents, etc. And there is also flexibility to do gynae as well as non-gynae slides. So if your laboratory is doing non-gynae slides, you can also process your non-gynae samples in here. And it also tells you when the reagent needs changing, when they're running out. So you don't run out of it in the middle of the batch. And when to also change the waste bottles. It prompts the user how to do that as well. And the workflow when you're using SlidePrep is once you have received the vial, you will then vortex it. Normally, laboratories use a multi-vortexer because the volume is high and the vortexer can accommodate more than one vial, as you can see here. And then you use a PrepMate for cell enrichment process, a PrepMate as well as centrifuge for cell enrichment process. And then the tubes that are obtained goes on to the SlidePrep for processing as well as staining. Once you've got the stained slide, we then cover slip it and then look at it under the microscope for diagnosis. So how is BD SurePath different? It's because we use the cell enrichment process and not filters. And as you can see here, the cell enrichment process removes most of the blood from the sample. And in the end, what you get is a solid cell pellet with diagnostic material. So the cell enrichment is proprietary to BD and it uses density gradient. Density gradient is always put in first and then the top layer will be the sample. And this minimizes obscuring of the diagnostic material, obscuring of your squamous or your endocervical cells, giving the clean background. It also reduces the unsatisfactory rate and increases the abnormal detection rates. And here's a Bigras study which tells you that about 37% of the cellular material can be lost when you discard the sampling device. And not only this, but also the endocervical cells, which are mucus-producing cells, sometimes they also slip into the, during the swirling process, they cannot come out easily onto the preservative fluid. So not only are you throwing away some of the squamous cells, but also some of the endocervical cells along with the sampling device. Another study by Sweeney shows you the benefit of the cell enrichment process. And in this graph, you can see that with the increasing intensity of the blood, your cellularity decreases. But with BD SurePath, the sample is still adequate as long as we use the cell enrichment process. However, compared to other LBCs, it may not be the same result. A simplified diagram of the workflow with 3 options offered by BD. The first one is mid-volume laboratories, where a sample comes in to the laboratory. It is then vortex using the multi-vortexer, and then it is put through the PrepMate, which performs the cell enrichment process, and afterwards it goes through the SlidePrep for processing and staining. Option 2, where the volumes are quite high, again, you vortex the sample and then you put it in the multi-processor. The multi-processor, it not only does the cell enrichment automatically, but it also centrifuges the sample inside the multi-processor, and all you get is the centrifuge tubes, which you can put into the SlidePrep here to process your slide. Now, the multiprocessor, you can also produce molecular tubes. So if you are doing HPV staining in your laboratory at the same time, with the same vial, you can do the C tube, which is for your LBC, and you can do M tube, which is for your molecular HPV test. And these M tubes goes onto the Viper LT system, which does the HPV screening. For mid-volume labs, you can also have Viper LT separately. So once the LBC processing is finished, you can then put the tubes for HPV screening. Now, the third option is semi-automated processing for low-volume laboratories, where, again, you vortex the sample for homogenization, and then you process it using the DTS method. Once your slide is made, if there is a need for HPV test, you can use the vial, which has the leftover sample, and you can process it in the Viper LT for HPV test. So our Viper LT, which performs HPV test, is a fully automated tabletop instrument. The throughput is 1 to 30 samples per run, or once the first 30 samples are under amplification, you can keep on putting more samples, so it gives you about 120 results in one single shift or in an 8-hour shift. And the reagents are ready to use, so you don't have to have a technician to prepare the reagents or anything. You just have to open up the reagent sample and then put it into the Viper LT, and they can all be stored in room temperature, so you don't have to have a refrigerator to store anything in colder environment. Viper LT is also compatible with the lab LIS system. Here's a workflow using Viper LT and BD SurePath vial. You only need 0.5 mils of the material, so you still have 9.5 mils left in the vial for other processing. And the 0.5 mils from the SurePath vial goes into the BD diluent tube, which is first heated for an hour. The heat step is 1 hour. And then the diluent tube is put into the Viper LT for extraction and amplification. And this takes 3.5 hours. And at the end, you receive a HPV report indicating whether the woman is HPV positive or HPV negative. If you're using any other LBC method, for example, thin prep or any other LBC method, you will require 1 ml of sample. It can also, we can also use Copan flock swab or any other self-sampling device. And my next slide will show you which other sampling devices is BD validated for. Here I have used a flock swab as an example for self-sampling. So once you receive a flock swab, you can then put it into the diluent tube, which goes for 1 hour heating process. And then after heating, the tube is put into the Viper LT system for 3.5 hours for automation and extraction and amplification. And then the HPV report indicating the HPV positive or HPV negative is generated. So the whole process takes 4.5 hours. And there are 3 simple steps to it. You transfer and then heat for 1 hour and then load it onto the Viper LT system. BD is also the first diagnostic company to offer self-sampling devices for BD Onclarity HPV assay. So this means that Evelyn brush, Viva brush, and flock swabs are all validated for Onclarity assay, and you don't need additional in-house validation. Most advantages is to target the undeserved population or population that is in rural areas. So that's the advantages of using self-sampling device. It is low cost and it can accelerate the transition to HPV primary testing, especially if you're transiting from LBC to HPV. And you don't need any liquid medium like in LBC, you need preservative fluid, but these are all dry swabs or dry self-sampling devices, and you don't need a alcohol-based material media for this. So just a little bit about BD Onclarity HPV assay. As you can see, it's a 3-well system. Each well has internal control, which is beta globulin, which ensures that DNA is present. And we do have individual reporting. So 16, 18, 45 is individually reported, and these are the high-risk HPV. Then there is regional variance, high-risk HPV, which is 33, 58 combined together, but 52 and 31 is individually reported. And then with lower risk HPV, 51 is individually reported, but the rest of them are grouped together. So in all, you have 9 results at the end from Onclarity assay. BD Onclarity assay is also FDA approved for ASCCP reflex, for co-testing, and for primary HPV screening. As well as it meets the Maya criteria for clinical sensitivity, specificity, and for reliable performance. So extended genotyping, why is it important? It allows risk stratification and better patient management. If you look at this diagram, the current screening algorithm is for high-risk HPV positive. For very high risk, it's referred to colposcopy, whereas the 12 other high risk are either goes back to routine screening or depending on their previous history, may not be referred to colposcopy. Whereas with extended genotyping, it helps for better management by risk stratification. So for very high risk, such as your 16, 18, or even 45, it is immediately referred to colposcopy. For high risk, which are regional variants such as 51 or 52 or 31, they are either retested in 1 year or again, depending on the previous history of the patient, can be referred to colposcopy. And for reduced high risk, which is the gray ones that I showed you in my previous slide, they can go back to regular screening and be monitored. So extended genotyping is also an advantage in a post-vaccination country if there is a post-vaccination program there. And to go into detail, value of extended genotyping is it offers risk stratification for better patient management. The results are available with HPV screening. So, for example, if the woman has come and you've done a HPV screening which has come back as high risk positive, but you want to know exactly what genotype it is, you don't have to call the woman again or repeat the test. You just have to go back to Viper LT and it unlock that genotyping result and it will tell you which genotype it is. It has also improved positive predictive value for women who have been referred to colposcopy. It reduces avoidable colposcopy for women who have been recommended for retesting. And you can also monitor women after 1 year by enhanced identification of true persistence follow-up of abnormal screening test. And this can also avoid unnecessary colposcopy. And if you're doing a test of cure after treatment, it can be enhanced by identification of true persistent, and that will avoid unnecessary retreatment. And just to share with you, one of the new instrument that BD has just launched recently is the BD COR System. And this is for more high volume laboratory, for centralized laboratories who have high volume for HPV as well as high volume for molecular assays. It is a modular system with a pre-analytical module in the middle where all the pre-analytical is done automatically. All you have to do is when you receive the sampling vials, be it SurePath or ThinPrep, all you have to do is feed it in through this hole here. And then if it's for HPV, it'll go to the GX module to test for HPV. This is where the amplification and extraction is done. Whereas if it's for other molecular assay like SARS-CoV or STIs, MGen, etc., it'll go to the molecular MX module, and it's all fully automated. So we have 6 to 8 hours walk away from the system. And the the GX model, the HPV model, has just been launched in Australia. In this region, there are lots of them already in the US and Europe. The MX is yet to launch in the world later this year. So just to share with you the latest addition to the BD solution to cervical cancer screening. And in conclusion, I'd like to also highlight that we have a robust team which is cross-functional across a lot of functions, which supports the cervical cancer screening in India. So we have our HEOR team or health economic team, which step in if you need to analyze the LBC as well as HPV models. We have the medical affairs team, which help in organizing the CME webinars and seminars. The applications team help in training. Technical and engineering team work together to provide a good service for the instruments. And then we have a sales team who provides all the information that your lab may require. So we all work together as one big team to provide the total solution for cervical cancer screening to you. And that is all from me. Thank you very much for listening. And if there are any questions, I will be happy